Hello, thank you and welcome to my presentation. I am Etourneau, a third-year PhD student at Grenoble-Alpes University, and I'm going to present you my work here, which is called Pirat, a peptide-level imputation method for randomly truncated proteomic data. So let's have a look first at about what is mass spectrometry-based proteomics data. Here is an example of such dataset. So it has few specificities. First, you can see that we do not directly quantify proteins, but instead we quantify fragments of proteins, which are called peptides. And because of that, some peptides can be shared between several proteins. Secondly, we are in a huge dimensionality, which means that we have thousands of peptides for usually quite few samples. And last but not least, there are missing values which are said missing not at random, or MNAR. And I'm going to tell you what that means and why it's an issue. So in proteomics, missing not at random values means that the abundance is missing because it is probably too low, and the mass spectrometer cannot detect the peptide. But there are also other missing abundances that can occur completely at random over the whole dataset, and that's why they are called missing completely at random. So we expect that the actual distribution of abundances, if they were all observed, so this corresponds to the red histogram here, well, this distribution should be different from the observed one that the instrument has actually measured, and the red one should be a bit left shifted compared to the blue one. To impute these missing abundances in very high dimensional setting and still leverage correlations between peptides, well, we have to lower the dimensionality of the problem. And to do so, we make groups of peptides based on proteins they belong to. So now each group can be imputed separately with a multivariate imputation algorithm, and the different results we have for shared peptides, for example peptide 2 and 3 here, well, we can simply average these results. So now we have faced the dimensionality issue, but not the presence of low abundance censoring, and this is what we are coming to. So Pirat is based on a multivariate likelihood model, and we simply aim at maximizing the joint likelihood of observed abundance values and the missingness response of value. We make a Gaussian assumption for the complete data, and we estimate its mean and covariance matrix by maximizing this quantity, this likelihood. However, to do so in MNAR setting, we have to define a missingness mechanism, which is roughly the probability for an entry to be missing, depending on its underlying value, and some parameters, gamma 0, gamma 1, which can be estimated from the whole peptide datasets with a simple regression. So once we have all of this, we can impute the missing values by their expectation with respect to the observed values here, the missingness response, and the estimated parameters. And we compute it by Monte Carlo method, and the job is done. So now, to see whether Pirat performs well, we evaluate our method on differential analysis task, which is closely related to biomarker discovery. And in proteomics, we have some benchmark datasets where we know which proteins or peptides should be found as differentially abundant across treatment groups. So all we have to do is take the dataset, apply our imputation method, then perform a statistical test for differential abundance, and one way or another, for example, and we can assess finally the prediction of differential abundance by the p-values obtained with a ROC curve, for example. So we repeated this procedure for 3 different datasets and compared Pirat with 15 other imputation methods. So we achieved the best AUCs on the 3 datasets and for a high specificity level, which corresponds to classical biomarker discovery setting. So it turns out the top scoring method varies dramatically with the chosen dataset. And this is probably representative of a varying MCAR-MNAR proportions. So it demonstrates that our method, Pirat, is robust to such variations in these proportions. In summary, we developed Pirat, an imputation method dedicated to peptide-level proteomics data. It leverages correlations of peptides between peptides from same proteins and automatically estimates a random left censoring mechanism to cope for MNAR values. And finally, we demonstrated its superiority to other methods for biomarker discovery tasks on 3 different datasets. So, thank you very much for watching until the end, and I would really be pleased to chat with you during the conference. You will probably find me next to the buffet if I'm not next to my poster. See you soon!